In the last video, we ended by talking about how pyruvate is considered an alpha amino acid because at this alpha carbon, the carbon right next to the carboxylic acid, there is a ketone attached to it, and it also has the carboxylic acid. So it is an alpha plus keto plus acid, an alpha keto acid. This is the simplest form of an alpha keto acid you could have. But if you remove this ketone group and switch it for an amino group, you're going to end up getting a molecule that looks like this, where you still have the same alpha carbon in the center right here. And now this alpha carbon has the same 4 attachments that the alpha carbon of an amino acid has, and it's in the same orientation. So you can switch an amino group for a ketone or vice versa and make an amino acid. You can tell which amino acid it's going to be because its R group is going to be right here. And in this case, this amino acid is alanine. So you could go back and forth between making pyruvate and alanine by moving an NH3 or the ketone. Now, pyruvate is just one keto acid. Let's talk about another amino acid that actually ends up showing up in the citric acid cycle that is called alpha ketoglutarate. Now, on alpha ketoglutarate, we can still identify that alpha carbon right here. That's where you've got a ketone attached, so it's still an alpha keto acid. But this one is not going to make alanine if we switch that ketone and make it a nitrogen group. Now, once again, we have made an amino acid where we have the R group. This R group has a carboxylic acid attached to it, so we know that it's an acidic amino acid. Glutamate has 2 carbons where aspartate has 1, so there's 2 carbons before the COOH means this is glutamic acid, or glutamate if you remove this hydrogen. If you don't remember the difference between ate and ic acid, check out part 1. But this specific part of the reaction is of the most importance because alpha ketoglutarate can enter the citric acid cycle and regenerate oxaloacetate, and glutamic acid can go on to be degraded into other things or can be recycled into other equations to make more alpha ketoglutarate. This amino group that's on a glutamic acid is going to be shuttled back and forth between different molecules, including alpha keto acids, to make amino acids and also make other molecules that can enter the citric acid cycle. So the general equation is going to be some alpha keto acid plus glutamate. It's going to create or be created by, depending on the situation, some other amino acid plus alpha ketoglutarate. So depending on what keto acid you've got is going to determine what amino acid you're going to make from that. But there's always going to be glutamate with the alpha ketoglutarate, which is going to enter the citric acid cycle. Now, moving an amino group takes a lot of energy and it creates a not so stable molecule. So the easiest way to do this is to be able to add some kind of enzyme. The first enzyme that we're going to be talking about and how its reaction occurs is aspartate transaminase. So like I mentioned previously, every reaction that involves glucogenic amino acids is going to involve glutamate plus some alpha keto acid. It's going to make an amino acid plus alpha ketoglutarate. In this case, the specific amino acid that we're making is aspartate. So the question is, if we're trying to make aspartate, and I know what aspartate structure looks like, what alpha keto acid would I have to insert in order to be able to make aspartate on the other side? You could do this 2 ways. You could either memorize the equation or you could actually figure it out. So if we draw out the structure of aspartate, so here's the structure of aspartate, and we already know the structure of alpha ketoglutarate. We got to switch this nitrogen with this ketone. And if we just draw them on the other side, we also already know what glutamate looks like. So now the question is figuring out what specific alpha keto acid is going to happen. We know it's going to look basically like aspartate, but instead of that nitrogen group being there, this is going to be another ketone group. from the alpha-ketoglutarate. This molecule is called oxaloacetate. And if you think back to how gluconeogenesis and glycolysis were different, you'll see oxaloacetate is one of the molecules that is able to enter gluconeogenesis, but is not involved in glycolysis. So it helps regulate gluconeogenesis. This using aspartic acid to make glutamate and making oxaloacetate from alpha-ketoglutarate, that's a really important reaction. And the enzyme that is specifically used to catalyze or make that reaction happen faster is called aspartate transaminase, which is often abbreviated to just be AST. The other key enzyme that is involved in making glucose from amino acids is called alanine transaminase. Now, like the name might suggest, instead of using aspartate or aspartic acid as the other molecule going with the glutamate, we're going to use alanine. So we're going to have alanine plus that alpha-ketoglutarate is going to make or be made by glutamate plus some alpha-keto acid. Once again, you could figure out what the alpha-keto acid is, but as we mentioned previously, we already know the structure of alanine, and we know that it specifically came from pyruvate. Pyruvate's another one of those molecules that goes straight into gluconeogenesis. Making pyruvate to be able to do gluconeogenesis is incredibly important. The amino acids involved in creating pyruvate and oxaloacetate are going to be very important. So we've got the alanine transaminase, which can be abbreviated to the ALT, and then we've also got the AST or the aspartate transaminase. So now that I've walked you through how to figure out which alpha-keto acid is going to be needed in order to make these amino acids, I've got a couple questions for you, and put your answers in the comments below.